Thanks for showing up on a Wednesday afternoon. This is our invited speaker seminar time, or additional seminar time. So it's my pleasure today to introduce to you Dr. Chuck Madenjian. Chuck and I don't count years anymore, but we've known each other for a long time. We figured out earlier today. And Chuck is a scientist with the USGS Great Lakes Science Center in Ann Arbor. Prior to that, Chuck, way back in time maybe, was at Rutgers University for his undergrad. He was at U of M for his master's, and then someplace in Hawaii, University of Hawaii for his PhD. And then Chuck spent a number of years working with Stephen Carpenter and Jim Kitchell at the University of Wisconsin at the Center for Limnology there. And he's widely known in the area of fish bioenergetics modeling. He's done a lot of work with fish contaminants in the last few years. He's worked with rainbow smelt, walleyes, just about everything. So with no further ado, I'm going to turn it over to Chuck.
Yeah,
help him.
I might need your assistance, Steve.
Right.
Thank you very much for that intro. Actually, the guy that's done the bioenergetics modeling is right here, Steve Chips. But I wanted to begin this talk with if we could, with a short but very insightful video with Dan Aykroyd and Wayne Newman.
How many times has this happened to you? You have a bass. You're trying to find an exciting new way to prepare it for dinner. You could scale the bass, remove the bass's tail, head, and bones, and serve the fish as you would any other fish dinner. But why bother now that you can use Robco's amazing new kitchen tool, the Super Bassomatic 76? Yes, fish eaters, the days of troublesome scaling, cutting, and gutting are over, because Super Bassomatic 76 is the tool that lets you use the whole bass with no fish waste without scaling, cutting, or gutting. Here's how it works. Catch a bass, remove the hook, and drop the bass. That's the whole bass into the Super Bassomatic 76. Now adjust the control dial so that that bass is blended just the way you like it. Yes, it's just that simple.
Wow, that's terrific bass.
We've got fish here fast and easy and ready to pour. Bassomatic 76 comes with 10 interchangeable rotors, a 9-month guarantee, and a booklet, 1001 Ways to Harness Bass. Super Bassomatic 76 works great on sunfish, perch sole, and other small aquatic creatures. Super Bassomatic 76, it's clean, simple, and after 5 or 10 fish, it gets to be quite a rush. Super Bassomatic 76, you'll never have to scale, cut, or gut again. Order again. That's Pier 25, New York, New York. Come on, Bassomatic.
Some of you may be too young to remember this skit. Some of you may not be, but
Yeah.
it was a classic.
Yeah. How many, was that the first time that you saw the Bassomatic? Wow. That's quite a few. Yeah, it came out in the 75 or 70. So, oh yeah, thanks again, Steve. So yeah, it's my pleasure to be here. I'm looking forward to presenting some of the information that we've obtained over the years. And so we've put a lot of fish through the Bassomatic, hundreds and hundreds, and the story has emerged. So my job here is to relate that story to you. And I would like you to pay attention to the whole fish, because the study I'm going to present today is based on whole fish contaminant concentration differences between the sexes. So the key is whole fish. And the reason I'm going to talk about whole fish contaminant concentrations is that's the only way that we can be absolutely certain that the concentrations that we're reporting reflect the entire fish rather than part of a fish, where we run the risk of coming up with differences based on a part that might not accurately reflect the entire fish. So we'll be talking about whole fish differences in contaminant concentrations between the sexes and the patterns that have emerged over the years. So this effort, I'm not the only one involved. Obviously, there are a load of people that have been involved. George Noguchi was the one that first got me started in this line of research over about 20 years ago. But there have been other people that have been generous with their time and thoughts, including people like Rick Radisky and Sergei Cherniak and Dave Krabbenhoff at the USGS lab, water science lab in Wisconsin as well. So just a brief background on polychlorinated biphenyls or PCBs. They were manufactured by Monsanto, used in electrical transformers, also to finish off wood products for a while, hydraulic fluids, used as a lubricant in industrial applications. It's a highly stable molecule. It doesn't break down readily in the environment. And so there was growing concerns in the 60s and 70s, health concerns, because it was a suspected carcinogen, and that prompted restrictions on use of PCBs beginning as early as 1970. And those restrictions were effective in that there was a dramatic decline noted in PCB concentrations of Great Lakes fishes beginning in the 1970s, so that those levels have come down over an order of magnitude, maybe closer to 2 orders of magnitude over the last decade due to the restrictions on use. A few more things about PCBs. The concentration in the fish is equal to what's called the body burden, PCB body burden, divided by the weight of the fish, where the body burden is just the weight of the chemical. So if you were able to pull out the chemical from the fish and weigh it, that would be referred to as the body burden. And you divide that number by the weight of the fish, and you now get the concentration in the fish, which was typically expressed in milligrams per kilogram or parts per million. But nowadays, with the concentrations being so low, it's typically expressed in parts per billion or nanograms per gram. And the gross growth efficiency of the fish is equal to the amount of growth shown by the fish over a period of time. That is the increase in the weight of the fish divided by the amount of food that was needed to attain that growth. And it can be shown that gross growth efficiency is inversely proportional to PCB concentration. And so the easiest way of picturing that is if you have 2 fish and you feed, all other factors being equal, you feed them the same amount of food, but one fish grows faster or grows more from that amount of food than the other, then the fish that grew more will have a lower concentration because that chemical is now being diluted out over a greater mass of the fish. This is referred to as a growth dilution effect. And so if we find that males are 20% higher in PCB concentration than females, then if all of that difference was explainable by by this growth dilution effect, that would mean that the females were 20% more, would have a 20% higher gross growth efficiency. That reflects the inverse relationship. So males higher in PCBs by 20%, then if the difference in gross growth efficiency between the sexes were to explain that, then the females would have to have a 20% higher gross growth efficiency. So our story begins in Saginaw Bay this afternoon, which is this large embayment of Lake Huron, the main tributary to which is the Saginaw River, which flows south to north here, and one of the tributaries is the Tittabawassee River here. So at spawning time, the Saginaw Bay walleye population ascends these 2 rivers and spawns in the Tittabawassee River in a stretch close to the Dow Chemical Company site. So these fish are actually spawning on Dow Chemical Company site. Now, Dow Chemical dumped a lot of PCBs into the Tittabawassee River in the 50s and 60s, and that chemical accumulated in the mouth of the lower Saginaw River, so much so that it was referred to as a PCB hotspot, in that the amount of chemical, or the concentration of the chemical in the sediments of the lower Saginaw, were orders of magnitude higher than the chemical concentration in the sediment in Saginaw Bay, or even more higher. And the sediment in the main basin was even lower than the sediment in Saginaw Bay. So as you moved away from the hotspot, much lower concentrations, exponential decline as you moved away from that hotspot in space. The spawning occurs in early April, typically, for the walleye population in Saginaw Bay. Just an underwater shot of walleyes. And what we noticed, if we sample those walleye from the spawning run, is this very curious pattern of males being about 2.5 times higher in PCB concentration than the females. The male concentrations are the blue triangles in this plot, and the females are the yellow circles. So you can see that the females average slightly higher than 3 parts per million during this particular sampling, and the males average all the way up to 8. So the males are about roughly 2.5 times higher in PCB concentrations than the females. And through a series of hypothesis testing, George Noguchi and I concluded that the main driver of this big difference in contaminant concentrations was that the males were spending quite a bit more time near the mouth of the Saginaw River, and therefore receiving a lot more PCBs in their food at the mouth of the river, and therefore having a much higher concentration. One of the main pieces of evidence for this conclusion was a data set gathered by the Michigan Department of Natural Resources, where they would go out every April and shot walleyes right on Dow Chemical property. You can see beautiful Dow Chemical Company in the background here. And then bring the shocked walleyes, bring those walleyes over to the shoreline to receive a jaw tag, a numbered jaw tag. And because the fish were getting close to spawning, they could be easily sexed. They were ripe and running, essentially. Each walleye got a jaw tag and then was released back in the river. And over the years, there have been 20,000 recaptures of the walleyes. And the angler would typically report the number and the site of the recapture. So what George and I did was we just divided up the region into 3 areas. One, the river and its tributaries, the Saginaw River and its tributaries. 2 was the Saginaw Bay proper, and then 3 were all other areas outside the river bay system. So that would include all of the main basin of Lake Huron, but some walleyes were actually recaptured in Lake Erie as well, Lake St. Clair, Detroit River, St. Clair River. And those results showed that over 60% of the male recaptures in the blue bars shown here were in the river or the river system, whereas only about 33% of the females. The bulk of the female recaptures were in Saginaw Bay proper, and if a walleye were to be caught outside the Bay River system, it was more likely to be a female than a male. So clear and significant differences in the habitat use between the sexes. And so, and we also know that the few measurements that have been made of the prey fish show considerably higher PCB concentrations in the river system than in the bay, and those concentrations would even be a little bit higher than concentrations of the prey fish in the main basin of Lake Huron. So again, very solid evidence for a spatial different habitat use between the sexes, and that was the main driver for this big difference. So we could ask the question, what would happen to the walleye PCB concentrations if we removed that hot spot? But again, to summarize, there were two conditions needed for this, what we call the hot spot effect. One, that there had to be a physical hotspot in the ecosystem. In other words, there had to be a hotspot for PCBs within the ecosystem occupied by the fish population. And two, that there had to be a difference between the habitat use between the sexes, where one of the sexes would occupy an area close to the hotspot and therefore receive elevated PCB concentrations in their diet, whereas the other sex would occupy an area further away from the hotspot and receive lower PCB concentrations in its diet. This combination of conditions doesn't occur a lot in nature. It would be in a relatively few amount of fish populations around the world, but if these two conditions are met, this hotspot effect is realized, and you get considerably higher PCB concentrations in one sex versus the other. So we do see that the conditions are met for Saginaw Bay walleye, but there's also a case of both conditions being met for Hudson River striped bass, the Hudson River, the big river going through New York City. So we can ask the question again, what happens if what would happen to those walleye differences in PCB concentration between the sexes if that hotspot was removed? And so to answer that question, we sampled walleye from South Manistee Lake here in the Upper Peninsula of Michigan, a lake which receives its only PCBs from atmospheric deposition. So there's no direct input of the chemical from some kind of point source. All of the chemical received in the lake is from atmospheric deposition, PCBs that come down from the atmosphere through rainfall or snowfall and end up in the lake. Again, this lake is located in the Upper Peninsula of Michigan, about 2 miles long by 1 mile wide. The deepest depth is about 30 feet. And if we look at those PCB concentrations, we see that the males on average are higher, but the difference isn't as dramatic as for the Saginaw Bay walleye. Comparing them side by side, the males from South Manistee Lake were about 34% higher in PCB concentrations on average, whereas the males in Saginaw Bay were 150%. So quite a dramatically higher difference. But nevertheless, the males are, with the hotspot removed, the males are higher in South Manistee Lake still, just a much smaller difference between the sexes. Well, we also noticed that for lake trout from Lake Ontario, males were higher than the females by about 22% at an age of about 5.5 here. So at the average, the general mean for the ages, males were 22% higher in PCB concentration compared with the females in this Lake Ontario population. And for coho salmon from Lake Michigan, we also found males higher in PCB concentration on average by 19%. So that the males were 19% higher in PCB concentration than the females. So if we formed a chart South Manistee Lake, 34% higher. Lake trout from Lake Ontario, 22% higher. Lake Michigan coho salmon, males higher by 19%. Now, in the South Manistee Lake walleye population, there was a definite difference in growth rates, with the females being considerably higher for ages 5 and 6. Females were about 50% higher in weight. So a definite difference in growth rates, with the females growing considerably higher. But in the other two populations, virtually identical growth rates between the sexes. So the males were growing just as fast as the females. One other thing I wanted to point out was that in the coho salmon population, the salmon were sampled prior to spawning. So the salmon had ascended the tributaries in Lake Michigan and were getting close to spawning, but all of the fish that were sampled were caught prior to spawning in that system. So we asked the question, what could be driving this slightly higher difference, the males being somewhat higher in PCB concentration than the female? And the first hypothesis we considered was it possible that the females were dropping their eggs at spawning, and there was so much PCB in the eggs that the concentration of the females would drop substantially, and that was the main driver of the difference in PCB concentrations between the sexes. That is, the females losing so much that their PCB concentrations dropped dramatically. Well, we already know that this hypothesis doesn't hold water, because if you recall, the coho salmon that we sampled were caught prior to spawning. And as we know from our biology courses, the coho salmon is a semoparous species. They spawn only once and die. And since these fish had never spawned before, the fish that were sampled had never spawned before in their life, this could not be the reason for the difference in the concentrations. But there is also other evidence that would discount this hypothesis. And that was evidence from when we do measure PCBs in eggs and the somatic tissue, that is the rest of the fish. So you would send the eggs through the basomatic, a little basomatic, and then send the rest of the fish through a grinder and determine those PCB concentrations. And weigh the eggs and weigh the somatic tissue or the rest of the fish. You can predict how much that female is going to change in PCB concentration after she drops her eggs. And you can see from this graph that in the majority of the cases, the female concentration would actually increase after spawning. And this is possible if the concentration of PCBs in the eggs is lower than the concentration in the rest of the fish. So if the fish ends up losing more of its weight than the body burden, and therefore its concentration actually goes up. Now, not a lot. You know, the highest it goes up is about 10%, but nevertheless, in most of these species, the concentration of the female is actually going up, and therefore there's no way that this factor could explain the males being higher in PCB concentration than the females. Yeah, there's a few species where it goes down, but in most species it actually goes up. So this hypothesis really doesn't seem to be the one responsible for the differences between the sexes that we saw for the three species. What about the growth dilution effect? Well, the growth dilution effect might explain the difference for walleye, but not the other two species. So to test this hypothesis, we did look at what the growth dilution effect could be doing for the walleye. And to do that, we used a bioenergetics model where we would fit the model to observe growth and then estimate the gross growth efficiency, divide the attained growth by the gross growth efficiency, or I'm sorry, divide the, we use the model to estimate food consumption. So we fit the model to observe growth to estimate food consumption, divided growth by food consumption to estimate gross growth efficiency, and then form the ratio of the female gross growth efficiency to the male gross growth efficiency. And here are the results of that analysis. The y-axis is the ratio of gross growth efficiency for females divided to the gross growth efficiency to males. And you can see that it levels off at about 10%. So the females were 10% more efficient at converting food into their growth. So this would account for males being 10% higher in PCB concentration compared with the females. But you'll recall that the males were 34% higher in PCB concentration compared to females. So this explained a portion of it, but the majority of the difference was unexplained by the dilution effect, growth dilution effect. So how about this effect of lipid concentration? Well, there was a time back in the 60s and 70s where people thought that lipid concentration drove PCB accumulation in fish. But we know from subsequent research that it really isn't lipid concentration that's driving the accumulation of PCBs. It's actually food consumption. The main intake of PCBs to a fish is food consumption. So it's not the lipid concentration in the fish that's driving the accumulation of PCBs. Rather, it's the food consumption by the fish and the amount of PCBs coming in through the diet that's driving PCB accumulation. So this idea that lipid concentration is doing it really didn't pan out. Yes, there is a correlate. If you were to plot PCB concentration against lipid concentration, you do get a general positive increase, but that's just a correlation, not the causation. I also would like to point out that in the walleyes from South Manistee Lake, the females were slightly higher in lipid concentration than the males, not much, but the lake trout, the males were slightly higher. But in all three cases, males were higher in PCB concentration. So this factor really doesn't explain the difference as well either, this hypothesis. What about the idea that males expend energy at a higher rate compared with females, both through more swimming activity and also even when the fish are resting, their metabolic rate is higher. Therefore, they are, to grow the same amount, they have to consume more food. Therefore, they then bring in the chemical, that is PCBs at a higher rate, and that's the reason for the higher PCB concentration than in the males than in the females. Well, there is, if you look through the literature, there isn't much. Most of the time, people don't bother to compare differences in swimming activity between males and females. They just want to measure the activity and they don't even consider a sex difference. But in the cases where people actually keep track of it, they've found that male activity is higher than female activity. Here's a case of Atlantic salmon in a Scottish river where they actually implanted heart rate monitors and found out that the heart rate of the males was significantly higher than females, and they concluded that the males were burning off energy at a higher rate. And another study on brook trout using pit tags where they documented much more movement across seasons in males compared with females. And then again, with the resting metabolic rate, in most cases people aren't looking for a sex difference, but when they do here's a study on brown trout where the resting metabolic rate of the males was 14% higher. So not a lot, but still a higher resting metabolic rate. Another for a marine fish, and then another study with a telemetry study for largemouth bass documenting a higher resting metabolic rate for males at spawning time compared with females. So in the few studies that are out there, there is support for this idea that there is a higher energy expenditure rate in males compared with females in fish. Well, I explained some of these ideas to a colleague of mine at the Hammond Bay Biological Station. He was skeptical, Tom Binder, but he had done a study on activity in mature sea lampreys, and what he did was implant these pit tags in the lampreys that could be sexed. His main objective was to determine how activity of lampreys changes with temperature. So he had lampreys in tanks. Here he is implanting the pit tag, and then he would release the lampreys in tanks, and he would have the antennae at 4 locations along the tank, and he would document or index movement by the lamprey moving from one field end of one antennae to another. And that would be considered one movement, and he would count up the movements for individual fish over the night, and that was the activity index for that fish. And again, he could sex them, and he said he would go back to his data set and see if there was any evidence for a difference in activity, and he did. He came back, he sent me an email a couple of days later, and he said, yeah, the males were higher. Here is the activity ratio. That is the mean activity index for males divided by mean activity index for females. And in 2 experiments, the males were higher. This experiment has been duplicated another 8 times with similar results. So pretty good evidence that the males are more active in the lampreys as well than the females. So in a synthesis paper, I proposed the idea that I think that if you were to go out and sample some more populations of fish, this would be a general characteristic. Males higher in PCBs than females, and this difference being driven by an activity difference between the sexes and a higher resting metabolic rate. So went out and decided to gather more data, do some more surveying, and sample some more fish populations. So we went out and sampled burbot from Lake Erie, and also burbot from Great Slave Lake, which is pictured here. This is Yellowknife Bay of Great Slave Lake with the northern lights in the background. Two folks going out and trying to catch burbot through the ice on Great Slave Lake. We also sampled sea lamprey from northern Lake Huron, so some lampreys out in the lake. Well, actually, they were on the spawning run. Sampled some lake herring or cisco from Lake Superior, lake whitefish from northern Lake Huron, and even some summer flounder from off of the Jersey coast. This is a summer flounder, which is what's called a left-eyed species of flatfish, where both of the eyes are on the left-hand side. There are other species with both of the eyes on the right-hand side of the fish. A predator caught in the coastal waters on the Atlantic coast. And they were worked up right, they were sent through and processed right at the Marine Field Station on the Jersey coast. Here's a shot of Olaf Jensen putting a summer flounder to a bassmatic here. And here are the results. So now 8 species of fish, and in all 8, males are higher in PCB concentration, ranging anywhere from as low as 17% higher for the sea lamprey, all the way up to 43% higher for the lake herring from Lake Superior and the summer flounder off the Jersey coast. So we then said, became interested in another contaminant, which is what's called a natural contaminant, that is mercury. Now it's been proposed that both PCBs and mercury are tracers of food consumption. That is, if you measure the amount of PCBs or mercury in a fish and make some other measurements, you can use that concentration in the fish to try to trace the food consumption, the amount of food being eaten, because with both chemicals, the main driver of accumulation is food intake. So they've both been touted as tracers of food consumption. And so in theory, the differences between the sexes that we saw for PCBs should also be true for mercury. So we went ahead and accumulated or assembled the data to see if this was true. Were the sex differences for PCBs identical to those for mercury? And what we found was no. Other than lamprey, where the males were 17% higher in PCBs and 16% higher, for all of the other species, the pattern just disappeared. In that, if we were to look at lake trout, males were 22% higher, but in PCBs, but only 8% higher in mercury. And in each of the other cases, the ratio of mercury concentration in males to mercury concentration in females was always lower than the ratio of PCB concentration in males to PCB concentrations in females. In fact, in an exaggerated case, we see that the males are 29% higher in PCB concentration, but the females are 22% higher in mercury concentration. So a really big swing for burbot between PCBs and mercury. And we asked the question, what could be causing this different pattern? And that led us to look through the literature to see what could be going on. Clearly the chemicals are processed differently between the fish. What could be going on between PCBs and mercury? And what we know from the literature is long-term elimination of PCBs is essentially zero. So the mercuries of PCBs, so the food is eaten, there's a period of digestion and processing the PCBs, but after a set of days, there's no further drop in the body burden. Yeah, some PCBs are dropped with eggs or milk, but other than that, after the PCBs are in the fish and there's this digestion, allowing the digestion process, the body burden remains flat. It doesn't change. Mercury is different. We know that we can actually measure elimination rate of mercury, even though it's a very slow rate. Mercury seems to be able to be eliminated by the fish, albeit at a slow rate. So maybe the difference between the two chemicals is males get rid of the mercury at a faster rate than the females, and therefore that ratio of mercury in males to females would be expected to be lower than the PCBs. That maybe this is different. It could be the difference explaining the difference between the two chemicals, explaining what happens to those ratios. Now, when I first started looking at this, we looked to see if there was any case where the mercury in elimination by males was higher than that by females in fish. And I couldn't find a single study in the literature documenting this. But in the medical literature, there are studies looking at the difference in mercury elimination in laboratory mice. And those laboratory mice studies were interesting in that when they measured the elimination rate in the urine, they found that when the mice were were juveniles, immature, there was no difference at all in elimination rate between the sexes. But as the mice got close to being mature, the male rate of elimination jumped up, so it was 3 times higher than that of the females. And they went one step further, castrated some males, noticed a drop in the elimination rate of the mature males, so it was back down at the level of the females, injected females with testosterone, and found that their rates would jump up to, so it was at the same level of the males. So they concluded, these Japanese researchers, that somehow or other testosterone was enhancing the rate of elimination of mercury by the mice. Well, the testosterone that you see that are in mammals like mice and humans, it has the exact same chemical structure as the testosterone in fish. Oh yeah, I did finally locate some data that for northern pike from experimental lakes area in Canada, even though it was a very small sample size, providing some evidence that males did get rid of the mercury at about double the rate. If you look at these exponents, the male exponent is double that of the females. So the males, in this case, were getting rid of mercury at double the rate of the females. But getting back to fish, the fish do have testosterone. They also have something, the males also have an androgen very closely related to testosterone that is unique to the teleost fishes, called 11-keto testosterone. So fish do have that, except for very primitive fish. And lampreys do not have testosterone or 11-keto testosterone. So maybe the androgens that are found in sea lamprey do not enhance mercury elimination rate, and therefore that would explain why the PCB ratio for lamprey was almost identical to the mercury ratio. But in higher fishes, with this, we think that 11-keto testosterone and testosterone enhance the elimination rate, and that explains the pattern that we saw in the table. And when I told these findings to Wei-Ming Lee, he was very excited because he's done the work on androgens in sea lamprey, and he's been claiming now for years that lampreys do not have androgens like the higher fishes. So there's been other work with relating testosterone levels in birds and mammals with the gonadosomatic index, which is simply, in this case, it's the weight of the testes divided by the weight of the animal, the total weight of the animal. So what's been found for birds and mammals anyway, is testosterone tends to increase with an increase in the relative size of the testes, or the gonadosomatic index. Although there's no literature at all for fish. But we do also know that when it comes to GSI of males, it's very tough to beat bourbon, of all things, where, if you read, this is a clipping from the Yellowknife newspaper, which is a small town on Great Slave Lake, and if you read the clipping, you'll see that the claim is that burbot have the largest relative testes size, or here in the articles it says testicles, but that's just for the layperson, of all of the vertebrates, and the average size for some burbot populations is 11% or so. So 11% of the body weight is wrapped up in the testes for burbot. In fact, Pete Cott sent me an email saying he caught one male burbot that had a GSI of 27%. So he put very, very impressive, and then in parentheses, and humbling. So anyway, so there is a practical application of this to some work with people from the Faroe Islands, which is located north of Scotland here, where some of the folks eat these long-fin pilot whales, which are loaded with mercury. And so it's been a concern, a health concern, and studied by people from around the world, just this one population on the Faroe Islands. And so computer models have been developed to track or predict mercury concentration in men and women from the Faroe Islands. And one of the assumptions made in some of these models is that men eliminate mercury at the same rate as women, but the problem with the model is it consistently overestimates mercury concentration in men, but not in women. So we think that what has to be done with the model is to increase the elimination rate for men so that you could get a match in what the model is saying for mercury for men with what's observed in the population on the Faroe Islands. So this kind of has opened up a whole bunch of potential research. One would be to continue to survey fish populations to see how prevalent this characteristic of higher PCB concentration in male fish compared with female fish, how prevalent that is around the world. And then maybe back that up with telemetry studies where possible, where you could actually go out and see if you can document differences in swimming activity between males and females. And then in the laboratory, try to document differences in resting metabolic rates of males versus females. And then also to eventually use all of this information to develop sex-specific bioenergetics models for fish. So there are bioenergetics models out there, and a lot of times they're good models. Professor Chips has used them a lot, and they're used without regard to sex because that's the question that you're interested in. But there might eventually be some uses where you want to know the differences between the sexes, and so this information could be used to develop sex-specific models. And then continue to survey fish populations around the world to see how prevalent this characteristic is of the ratio of mercury concentration in males to mercury concentrations in females being considerably less than the PCB concentrations in males to PCB concentrations in females. Again, we think this difference is driven by a difference in mercury elimination rate between the sexes. And then actually going out and measuring mercury elimination rates in fish species to see how much it varies from one species to the other, especially for the males, because there could be big differences between the males, partly due to testosterone levels, but partly due to other factors as well. And then go ahead and use this information to refine the mercury mass balance models that are out there. The Canadians are very big on using mercury mass balance models to estimate food consumption by fish. Well, we think these models will need to be refined on a species-by-species basis, maybe, if possible, to reflect differences in elimination rates of male from one species to another, because the data in that chart definitely suggests that in some species, males can get rid of mercury at a considerably higher rate than others. And so so there's a need to go ahead and refine those models and go ahead and do research to determine exactly how these androgens like testosterone and 11-ketotestosterone enhance elimination rate in males. Try to explore reasons why the androgens that are in lamprey do not apparently do not enhance elimination rate. And then go ahead and develop a relationship between testosterone and GSI, just like we saw with birds and mammals. So in conclusion, this studying of the differences in contaminant concentrations between the sexes, both for mercury and PCBs, within and across species, has yielded, brand new insights in differences in physiology and behavior between the sexes, and that these differences that we see with fish may well apply to higher vertebrates. So what we're seeing with fish could very well apply to higher vertebrates as well. And that in some cases, and this would be, I think, the minority of fish populations out there, that there is this that there is a spatial separation of sexes at least part of the year, and we saw that with Saginaw Bay walleye and Hudson River striped bass as well. And then what this research does point to is males expend energy at a higher rate than females due to higher activity and a higher resting metabolic rate, and therefore are consuming food at a higher rate. Males, the data suggests that males eliminate mercury at a higher rate, and this higher elimination rate appears to be associated with certain androgens in males. And then, if we remember what Dan Aykroyd said at the beginning of the talk, that the bassomatic works great on small aquatic creatures, we think that this line of research on what we're learning about fish, because they're amenable to this going through the bassomatic, that we could learn something about higher vertebrates as well. So with that, I thank you very much for your attention, and I'll be glad to answer questions.
Yeah, we have a few minutes for questions. Let me turn the lights on.
Yeah. All right.
Hey, questions for Chuck?
Yeah, Mike.
I was just going to flip the coin here. Has anybody looked at the effects of estrogen and some of those in terms of conferring retention of mercury?
Yeah, that good question.
Because females have 11 ketotestosterone as well as estrogen, not the testosterone, so and there could be some interaction there.
Yeah, the estrogen question is an interesting one, and I'm not sure anybody's even looked at that yet. It wasn't done with the laboratory mice, I don't think, but some people have now suggested trying just that with fish, estrogen, and seeing if it enhances the accumulation of mercury. Yeah, good question. Yeah. Yes, Steve.
Finally, something for men, right? We die younger, we're more stressed out, but we can
We can get rid of mercury a little bit faster.
But my real question, Chuck, was so with some work we've done with walleyes and mercury, we see the same patterns.
Oh, no kidding.
With mercury in terms of the males having higher concentrations.
Yeah.
I attributed it to the growth difference.
Yeah.
But I never calculated the GE, growth efficiency, that way.
Yeah.
So I'm going to do that. We might be able to fill in your one table where you have NA.
Yeah, yeah.
Because we probably have
Right. Yeah. And I've done it now for a few walleye populations, and it seems like walleye's a pretty substantial difference in growth rate between the sexes. Where females are up, in the range of 50% bigger at a given age. You get up to the 5s and 6s and 7s, and so. And even with that big difference, though, it only explains about 10%. Now, I forgot to mention with the summer flounder that the difference in growth rates between the sexes is even more dramatic. The females are about twice as big by age 4. And so, but even then, when I go through the calculations, it still will only explain about an 18%, gross growth efficiency difference. So that 43% difference about 18 to 19% of it was from the difference in growth rates, still leaving the bulk of it from an energy expenditure difference between the sexes.
Any other questions for Chuck?
This body burden depends on the size of the fishes also. It would be varying with the size of fishes, other than male and female sexes.
Oh, yeah.
It could influence with the younger ones, maybe faster depositing.
Yeah, you're talking about the contaminant concentrations in the fish?
Yeah.
Yeah, we definitely, we take that into account. If you noticed in the lake trout, you definitely saw an increasing pattern for both sexes with age concentration definitely increased. But in quite a few cases, we've noticed now that there isn't any appreciable increase in the concentration with age in several fish populations. So I think that all depends on the diet composition schedule. That has an effect, and so in some cases, again, very strong pattern with age. Others not that strong. But all of that is taken into account in comparing between the sexes, the concentration between the sexes, the size of it.
Another one, general.
Well, we mainly do it by age. It's mainly an age-based, so we're looking at a particular age to do the comparison. Of course, in the case where it doesn't change that much with age, it really doesn't matter. You grab an overall mean and there's your comparison. You're taking an overall mean for the males, an overall mean for the females, and boom, that's your comparison.
Another one, Another one, general curiosity, that so much research on mercury poison and PCB concentration, all that, it's happening this side, this western hemisphere, this side. But in our part also it must be there, and we don't have much focus, or I don't know why there is no studies and there is no much interest. Of course, there may be a different outweighing interest there, but here it is more focused and more studies in this part on mercury poison of aquatic ecosystem, fish, all that.
Yeah.
And is there any specific reason that here it's more concentration is there, or that side it could be less, or what is that? General, out of interest.
You're talking about in the U.S.?
Yeah, yeah.
Well, I think with the formation of some of these environmental agencies like EPA in studying it very much from health concerns, that's prompted that even the states now have issued consumption advisories on fish. Now, of course, those aren't uniform. They can vary from one state to another. I'm sure South Dakota has its consumption advisories.
Yeah.
But the mercury is one of the big concerns with PCBs. It's a suspected carcinogen. Mercury. One of the big concerns with mercury is the developing fetus, for humans, is very, the brain development is very susceptible to mercury. So the big concern there is to issue warnings for pregnant women. Stay away, because for whatever reason, when the brain is developing in the fetal stages, it's very sensitive to mercury. Whereas, once you're born, it's no longer much of a concern at all, but for the developing fetus, it's a very much a concern. And then there are the sublethal effects. I just heard one autoimmune system. I think there's a sublethal effect from mercury too. But the one for the developing fetus, brain development is very much a concern, and I think that prompts a lot of the mercury advisories.
I was just curious about the differential allocation of the mercury or the PCBs in the females, like in terms of ovaries or in egg production. I was wondering.
Very, very good question, and I'm a lot of times I'm asked about that. Did you take into account the obviously, we know that the chemicals are stored differently. The PCBs are in lipid, almost always stored in some kind of lipid deposit or tissue, whereas mercury binds with protein. So, when I showed that one plot about the predicted change in female PCB concentration after dropping the eggs. We could also do that with mercury. And what you would see in there is the concentration of mercury in the eggs is always considerably lower than the mercury in the rest of the fish. So it's binding with protein, and it's in the muscle, it's in the muscle tissue big time. So that's another thing about it. But I still think that regardless of where it's stored, mercury is still touted as a tracer of food consumption. So both of the contaminants are coming in through the diet. It's just a question of that in the case of mercury, there is a considerable difference in the rate of elimination. In PCBs, no. But you're right that they're stored differently. If you were to in the fillet, there's not much you can do with regard to mercury. It's in there. It's right in the muscle in the fillet. PCBs, if you trim fat away from the fillet, like belly fat, you will be lowering your intake of PCBs, because if you trim away the fat, there goes a lot of the PCBs. So if you were to measure whole fish PCB concentrations, for some species, there isn't much difference between whole fish and fillet. But for a walleye, a lean fish, the concentration in the fillet might be 4 times lower than the whole fish. Perch, it might be 10 times lower than the whole fish. Of course, lake trout, with the fat in between the myomeres, the concentration in the fillet might be almost the same as the whole fish. So, whereas mercury, the concentration in the fillet might be a little bit higher than the whole fish, because the mercury is right there in the muscle. And in some species, the concentration in the fillet might be 2 times higher than the whole fish. So there is a difference in where it's stored for sure. Yeah. Like a bluegill, I've got some bluegill measurements where the concentration in the fillet is 2 to 3 times higher than the whole fish for mercury. Yeah, interesting way of how it's stored.
Any other questions for Chuck this afternoon? Well, Skip took another round of applause. A couple of quick announcements too. If you'd like to talk with Chuck some more this evening, we're going to go over to Wooden Legs if you're old enough to drink iced tea with us. And you can ask Chuck about his Ayacook process with dead flounder. That was a good story today. But we're going to be there about 6 o'clock. And right now we're going to meet with some students. There's 179 AFS students and others. If you want to join us here, student, feel free to come over and chat with Chuck for a little bit. Great. So thanks everyone for attending this afternoon.